Hello everyone, Mr. Linder here. Let's start Unit 2 of Physiology by introducing the endocrine system. So the endocrine system is really a set of tissues and organs throughout the body that produce chemical messengers called hormones. Hormones are secreted predominantly into the bloodstream by these specialized cells found throughout the body, and they typically regulate ongoing long-term function. The endocrine system is different from the nervous system in that the endocrine system takes longer to activate. So when we looked at Unit 1 and we focused on the nervous system, we were really focused on milliseconds of time and how quickly we could send sensory and motor information throughout the body to regulate physiological activities. The endocrine system is also a regulatory system. It just takes longer for the mechanism of actions to take place. It can be minutes to hours before we see an actual response from the endocrine system. So the endocrine system is not dependent upon the speed that's necessary in the nervous system to generate physiological responses. So typically when we look at hormones and we look at endocrine control, we're looking at things that regulate growth, development, metabolism, water balance, temperature, ion regulation, reproductive function, things that can take longer in time to elicit a response as compared to the nervous system, where we need this sort of reflexive response, where things need to be extremely fast and happen within milliseconds of time. But there's a lot of similarity between the nervous system and the endocrine system. And so the endocrine system is a great place to start Unit 2 because it is a control system in the human body. So how do hormones work? Well, they actually work similarly to what we see happening with neurotransmitters in the nervous system. Hormones act on target cells. And they act on target cells to control enzymatic reactions. They act on target cells to control ion or molecular transport. They act on target cells to control gene expression or protein synthesis. And they do that by predominantly binding to receptors, either in the plasma membrane or binding to receptors that are internal within the cell, and then eliciting a cellular mechanism of action. The nervous system uses chemicals, predominantly what we call neurotransmitters, to bind to receptors and elicit a response. And so we see a lot of similarity between nervous and endocrine system control. Hormones have been studied for a long time. If you go back and you look throughout history, this is just an example of some ancient art. Hormones have been obviously affecting humans since our existence, and looking at abnormalities or changes in the body is really nothing new. This artifact actually depicts something known as a goiter. And so you can see in the art here, there is this depiction of a large mass in the neck region, and that would represent a thyroid gland malfunction. And so this is known as a goiter. And we'll talk more about that in future videos. But studying hormones and depicting the pathophysiology has been around for a long time. If you look back in time through endocrine research, there really is some classical identification methods that were used by many researchers to look at hormone and hormone function and pathophysiology. The three main things that you can consider when you look at historical evidence for hormones and pathophysiology that relates to hormones would be, number one, looking at removing a gland. So researchers historically would remove glands from organisms and then look to see what happens to the organism when there's a deficiency. And so if you remove a gland, you'll get a hormone deficiency, so the hormone levels will decrease, and you will get some sort of pathology associated with that. Now, if you look at historical research, it's not that the researchers knew what hormone it was or what the mechanism of action was, but you could take out a thyroid, and from the evidence, from the data, you could determine that there is some sort of chemical from that gland that's going to be regulating mechanisms within the organism. And so if you took that gland out, you would begin to see problems. Perhaps you would see muscle weakness or fatigue, or perhaps you would see reproductive issues if you were removing ovaries or testes. And so this removal of glands from an animal, from an organism, was a primary way to do classical types of research. The second thing you could do is you could replace. So you could actually replace hormones. Again, without knowing what the chemicals are and not having names for these things, you could still replace the fluids, the extracts from the gland that was removed, and see if it would alleviate the pathophysiology. So for example, if a particular gland was removed, it could be ground up and then the liquid portion could be inserted back into the animal to see if the pathology would go away. And what that allowed the researchers to determine was that there's something in the juices, something in the extract that you're putting back into the animal that is regulating their function. And of course, now we know that the extract contained specific hormones. And those hormones are regulating, again, whether it's ion balance or molecular balance, like sugar balance, whether it was hormones regulating reproductive cycle and so forth. So nowadays we have a lot more information because we have an idea of what hormones are and we understand mechanisms of action better. But these are classical ways to determine that hormones existed and hormones are regulating what's happening in the organism. And the third thing from classical research that you could take a look at is creating a hormone excess. So by basically leaving a gland, but removing a gland from another animal, grinding up the organ, creating an extract, and then giving that extract to an animal, to an organism that already still has its normal functioning gland. So creating an excess was another way to study what hormones actually do. So the hormone is not in a deficit now. The hormone has increased. You could think of this as a hypersecretion pathology, where you have too much of a particular hormone, you have too much thyroid hormone, or you have too much insulin, or too much testosterone, et cetera, et cetera. And you could look at the pathophysiology symptoms that are associated with having an excess. So this would be your hyper situation. And of course, the first one would be your hypo. When you're talking about removing the gland and having the hormone decrease, that would be your hyposecretion, and you could look at the pathophysiology symptoms associated with that. Once glands were determined to produce hormones, then you could start going through the process of actually identifying what those hormones actually are, and then you can start looking at what their actual functions are. As we fast forward to today, we have lots of hormones to study. There are lots of classical type hormones, those hormones that come essentially originally from places like the thyroid gland, hormones from the pituitary gland, things that fall under this sort of classical hormone situation where you have a particular gland within the human organism, and it strictly produces hormones to regulate some physiological function. It doesn't have other functions within the body. The pituitary is designed to produce hormones. The thyroid gland is designed to produce hormones. Thymus gland producing hormones. If you look at adrenal glands producing hormones, okay, these really fall into sort of the classical definition of a gland within the endocrine system that produces hormones. But if you study this table closely, you'll notice that there's lots of other organs mentioned that have other functions and fit into other physiological systems, but they too can produce hormones as well. So you may notice that the heart is on this endocrine list. We know that the heart is part of the cardiovascular system and it's designed to pump blood, but it actually produces a hormone. The liver is on this list, lots of functions of the liver, but we do know that there is a relationship to endocrine function with the liver. Pancreas producing digestive enzymes, but of course it has an endocrine role as well. Insulin, glucagon. Kidneys, part of the urinary system, but it produces things like erythropoietin, which controls red blood cell production. So the point is that the definition of the endocrine system is expanding because it's not just classical endocrine organs anymore. We're starting to see, and we know, that lots of organs of the body produce hormones that can have regulatory physiological functionality. So what makes a chemical a hormone then? That's an interesting question as well. The definition of hormone has been changing over the years. The traditional definition of a hormone is really a chemical secreted by a cell or a group of cells. It's dumped into the bloodstream and it's being transported out to distant targets within the body. And also that the hormone is produced at very low concentrations. So you've got sort of this classical strict definition of a hormone. It's got to be low concentration, it's got to travel throughout the body, through the bloodstream, and that is our definition of a hormone. Turns out, though, that there's lots of other chemicals that are produced within the body that don't necessarily follow this strict definition. So I want to look at some of the exceptions to the rule as you look at this sort of classical definition of hormones. So the classical definition says hormones are secreted by a cell or a group of cells, and they're supposed to be this sort of isolated endocrine cells. But it turns out that the endocrine system obviously is bigger than we thought. You have adipose tissue, you have neurons producing hormones, and so our definition is expanding, that it's not just isolated endocrine cells. You can have neurons producing hormones, you can have adipocytes producing hormones. So traditionally, endocrinology really focused on discrete epithelial tissue that produced hormones, but now we're expanding it into connective tissues, we're expanding it into nervous tissue, and we're looking at how other tissues of the body produce hormones as well, not just epithelial cells. Because the endocrine system is supposed to be derived of epithelial tissue. But it turns out neurons produce neurohormones and immune system cells produce cytokine chemicals that regulate what's happening with the immune system, etc., etc. Hormones are strictly secreted into the blood. Again, that portion of the definition is expanding. Traditionally, we look at hormones circulating through the cardiovascular system and exerting their effects. But it turns out that there are chemicals called pheromones. Pheromones are essentially ectohormones. These are chemicals released by the organism out into the environment to elicit a physiological or a behavioral response within the species. So you can look at other organisms besides humans. You can look at ants releasing chemicals, or you can look at sea anemones releasing chemicals. You can look at various species up and down the animal kingdom using chemicals through reproductive behavior, mating purposes. And so it turns out that humans aren't any different. When they've done studies on humans, especially looking at, say, axillary sweat glands, it turns out that volatile steroids are being produced. So these pheromone chemicals are being released, and they can affect reproductive cycles in females. You may be aware that the menstrual cycles of females that live together can actually sync together, and that's due to pheromone release, chemicals being released from the axillary sweat glands. There are essentially pheromones that will attract you to the opposite sex. And so there have been tests where females will smell t-shirts of various individuals, and they'll rank them based on how attractive they would be to that particular individual based solely on the smell of their t-shirt. But what is it really? Really, it's the pheromones. It's the steroid chemicals. And so you may or may not be attracted to somebody just based on chemical signaling. Hormones are transported to distant targets. That's not always true. Sometimes hormones will exert their effects in closer proximity. So the traditional definition is that hormones are dumped into the bloodstream, and then they're going to circulate to other places within the body, bind to receptors, and exert their effects. But it turns out that there are lots of other chemicals. There are lots of growth factors, eicosanoid type chemicals that can act in what we call autocrine or paracrine regulation. Essentially, autocrine means to affect the same cell type. So you might have an epithelial cell affecting another epithelial cell, or a connective tissue cell affecting another connective tissue cell. But you also might have paracrine signaling, where you have one tissue affecting another tissue type, but they can be in close proximity to each other. And so the definition essentially becomes sort of a gray area, trying to define what's a hormone and what's not a hormone. So a lot of these autocrine / paracrine regulators, maybe they're not classified as hormones yet, but they could be classified in the future. You just need to understand that there are lots of chemical signals within the human organism, and they don't necessarily follow the traditional definition of a hormone. You'll see as you go throughout physiology, the same chemical exerting different effects based on where it's located in the body. So there's a chemical called cholecystokinin, which has neural functions, but it also has digestive functions, so in the digestive system, okay, affecting the stomach. So you can see variations of the same chemical just based on where it's located within the body. And so that tends to complicate definitions of things as well. Hormones exert their effects in low concentrations. That's not always true either. Traditionally, hormones have been measured at nanomolar or picomolar concentrations, and so they exert their effects at very low concentrations. But it turns out that not all chemicals do that. There's actually a molecule called histamine. You're probably most familiar with histamine as it relates to allergies, but histamine is also used within the digestive system. to assist in regulating acid production, stomach acid production. And so it turns out that it works at higher concentrations within the digestive system. And so it doesn't meet that strict definition of working at low concentrations like most traditional hormones do. And so the boundary between what's a hormone and what's a non-hormonal chemical signal has really become blurred just by finding exceptions to the rules. And then there's lots of, again, these cytokine chemicals that perhaps wouldn't even meet the definition of a hormone nowadays, but they were called a hormone traditionally. There's actually a chemical called EPO. That stands for erythropoietin. That's the one from the kidney. That probably would not meet the definition of a hormone today from strict standards, but it was called a hormone historically a long time ago, and so we just continue to call it a hormone even today, when it really might just fall into a cytokine category, which cytokine just means cellular chemical. So the point is that it's hard to define what a hormone actually is. So I'm okay when you study the endocrine system to just look at all of these different things as being hormones, and we can add to the list when we come across other cytokine chemicals. So other growth factors, other eicosanoid chemicals, and really we could probably treat them as hormones as well, because the list of hormones is expanding within the body. What can we say about chemical signals in general then? Well, we know hormones, we know chemical signals act by binding to receptors. And that's kind of a key point. That's something that we looked at in the nervous system. Neurotransmitters exert their effects by binding to a receptor. So that neurotransmitter is going to bind to a receptor and start its mechanism of action. Well, hormones do the same thing. Hormones are going to bind to receptors as well. Now, that doesn't mean you're going to get the same response everywhere, just like neurotransmitters can have different responses depending upon the type of receptor that it binds to. The same thing can happen with hormones as well. Depending upon the receptor location, you can have varying types of responses from the same hormone. So the response of a cell is known as the cellular mechanism of action. When a hormone binds the receptor, there's going to be a cellular mechanism of action. Cellular mechanism of action. And we'll look at lots of different examples in future videos of how when a hormone binds a receptor, you can get a response by the cell. Hormones can act on multiple tissues, so you can see one hormone affecting lots of different places of the body. As long as there's receptors in those locations, you're going to get a response. But the response can be different depending upon, again, location, point in time in development. There are some hormones that affect the embryonic tissues differently than the adult tissues. And so it really depends on a lot of different factors when you're trying to figure out what a hormone actually does. Insulin is actually a good example of giving you varied responses within the human body. If you take a look at this diagram, it just gives you some insight into how you can get varying responses depending upon how a cell responds to a particular hormone. So we have two situations here. We have the fasted state, basically when you're not consuming food and you're living off of the body's resources. And then you have the fed state, which is when you're introducing food into the environment and you're changing the blood concentration of nutrients. And then we have different tissues responding. We have adipose and muscle tissue responding, and then we have liver cells, liver hepatocytes responding. And so what you'll find is that the responses by different cells can be different to the same hormone. So if we're just looking at insulin, you can have different types of responses just based on where receptors are located and what organs you're actually talking about. So muscle tissue and adipose tissue show changes in glucose transport proteins as well as enzymatic changes when you look at overall glucose metabolism. So for example, in the absence of insulin, so when you're in the fasted state and you don't have insulin, when you look at adipose and skeletal muscle, you'll notice that there's no hormone attached to the receptor. So here's the insulin receptor, and there's no hormone attached to it. So you'll notice that the plasma membrane doesn't have any transporters inserted in the membrane. And so the glucose, right, is going to have a difficult time getting into the cell. So in this fasted state without insulin, you don't have these glucose transporters. And then internally inside of the cell, it affects the enzymes that we use for glycolysis in the Krebs cycle as well. In the fed state, when you've introduced sugars into the bloodstream and insulin gets released, so here we have the insulin hormone attached to the receptor, that's going to trigger a cellular mechanism of action that's going to allow for the insertion of transporters into the plasma membrane of the cell. And so now glucose can diffuse into the cell, and then you can utilize the enzymes within the cell to process the sugars and generate ATP. So depending upon whether you're in the fasted state or the fed state and the presence or absence of insulin, that's going to affect the transporters and the utilization of enzymes for making ATP when you look at adipose and skeletal muscle. That's not true when you talk about liver hepatocytes. When you look at liver hepatocytes, you'll notice in the fasted state, there are already transporters in the membrane. So in the liver, insulin is not affecting the glucose transporters, the GLUT transporters. It is affecting the enzymatic activity. Okay, so it's actually showing in this diagram, this portion over here, we are seeing changes in the enzymes and the production of ATP. So that is the same. But what we're not seeing is the changes in the GLUT transporters. Notice there are GLUT transporters in the fasted state, and there are GLUT transporters in the fed state. What's different then? And why do liver hepatocytes have GLUT transporters in both situations? When you're in the fasted state, you want glucose to be able to leave liver cells and go out into the bloodstream so that you can keep your blood sugar in homeostasis, right? You want blood sugar to be near the set point. And so we use our reserves that are found in our liver, and we break down the glycogen that's in the liver cells. We produce free glucose, and then that glucose can diffuse out because there are transporters already in the plasma membrane, and that has nothing to do with needing insulin. So the liver cells don't need to respond to the insulin. But in the fed state, they will respond to insulin. So here we have insulin hormone binding to a receptor on the liver hepatocyte cells, and they will respond by increasing enzymatic activity so that you can generate ATP in the liver hepatocyte cells, but you can also go on to store the extra glucose as glycogen. And you'll notice the direction of diffusion now is into the cells. So when you have excess sugars, we can bring it into liver cells and we can store it and we can use it to make energy. But when we don't have excess and we're in the fasted state, we can allow that glucose to leave the cells. So insulin's effects are different, same receptors, but different locations in the body. And so you're getting different responses. In the brain, insulin does not have an effect on glucose metabolism. The neurons, the brain cells, don't rely on insulin for metabolism of glucose. And so they have access to the sugar whenever they need it. Hormones also have to be terminated. They have to be shut off, just like what you see with neurotransmitters. So just like other chemical signals, hormones are going to have a limited duration within the body. The body typically avoids long duration of hormones by really doing a couple things. We limit the secretion of hormones, and then we also have removal or inactivation of hormones, whether it's in the bloodstream or whether by terminating the action of the hormone at the cellular level. Hormones can be degraded by enzymes. These enzymes can be found in your blood, but they also can be found in the liver. They can be found in your kidneys. And then the metabolites of the hormones can be excreted. The rate of hormone degradation is known as the half-life. So if you have a certain amount of hormone at a particular concentration, let's say we're at 10 nanomolar concentration, and you begin to degrade that hormone. So now we have less of the hormone. Now we're down to 5 nanomolar concentration. The time that it takes to reduce the concentration by half is known as the half-life. And so you can look up the half-life of hormones and see how long it takes to degrade them. Some hormones are going to last longer in the bloodstream than others, depending upon what the half-life actually is. Hormones that are attached to cell membranes, and again, hormones that are circulating in the blood can be broken down by enzymes that are present in the bloodstream and that actually seep out into the interstitial spaces and begin to degrade those hormones. So there's a mechanism for terminating hormone activity, just like there's a mechanism for turning off neurotransmitters as well. How do we classify hormones? The classification of hormones can be complicated. There's lots of different categories that we could put hormones into. One of the ways that we classify hormones is just by what they are made of. So we have categories like peptide hormones, so made of amino acids, but particularly lots of amino acids, so amino acids linked together. So you might call these peptide hormones, or we can call them protein hormones made of lots of amino acids. Then we have our steroid hormones. These are going to be hormones derived from cholesterol. These are going to be your testosterone, your estrogens, your estradiol, your progesterone, cortisol, lots of different steroid hormones, but they all have the commonality that they're derived from cholesterol. And then you have amino acid type hormones, so derived from single amino acids. So whether it's a tyrosine derived amino acid based hormone, or it's a tryptophan derived amino acid based hormone, that's going to fall into the amine category. So think of those as like single type amino acid hormones. The other way you can classify though hormones, maybe a little bit easier, is just looking at water soluble versus fat soluble. So if a hormone is soluble in water, then it's going to be on this side of the table. So we have things like follicle stimulating hormone, luteinizing hormone, insulin, antidiuretic hormone, growth hormone, etc., etc. These are all water soluble type, which should be familiar, especially like insulin. You know, the insulin has to be injected because it's water soluble. It can't just pass through your skin. It has to get into the bloodstream through an injection. So it's a water soluble. Water soluble hormones, what's interesting about them is they are going to predominantly attach to receptors that are found within the plasma membrane of a cell. So if you have a cell these water-soluble hormones that are circulating through the blood are going to get out into the interstitial space, and then those hormones are going to bind to receptors that are found in the plasma membrane. And then they're going to exert their cellular mechanism of action, which again we'll look at in future videos. If it's a fat-soluble hormone, these are going to be predominantly your steroids. Those derive from cholesterol. You'll notice that there are some on the fence right here. We'll come back to those. But things like testosterone, your estradiols, estrogens, progesterone, cortisol. What's interesting about these is because they're fat soluble, for one, they can actually be absorbed through your skin. That's why you can wear, for example, birth control patches. You can absorb estrogens and progesterone through the skin, and then it'll get into the bloodstream and circulate. But these particular hormones, for one thing, when they're in the blood, because they're not water soluble, they're going to have to be attached to something else. So they're going to need a carrier protein to help them circulate through the blood. And then when they get out into the interstitial spaces and they get to a target cell, they predominantly pass through the plasma membrane because the plasma membrane is made up of lipids. And so these hormones are going to get into the inside of the cell, and they're going to look for receptors that are found within the cell. So you have different mechanisms of action. So then they'll exert their cellular mechanism of action, but they'll do that predominantly within the cell. That doesn't mean that hormones like testosterone or an estrogen or a cortisol can't bind to a receptor in the membrane. They certainly could, but that's not the main mechanism. The main mechanism is to pass through the plasma membranes and get to the inside of the cell and exert their effects. And then, of course, you have these on the fence here. So you have thyroid hormones, T3 and T4. They're going to act like a fat soluble, even though they're not a steroid. Okay, these are amine-based. And these are amine-based hormones, but they're acting fat soluble. You have melatonin, which is, again, slightly fat soluble and water soluble. And so, again, it's kind of on the fence as far as its cellular mechanism of action. And so there can be some variations, some exceptions to the rules. But the main categories would be water soluble versus fat soluble, and really thinking about how they exert their effects on cells. Does the hormone bind to a receptor in the plasma membrane? Or does the hormone pass through the plasma membrane and bind to receptors inside of the cell? The last thing to look at in terms of classification is sort of naming some hormones. And so what you'll see when you look at peptide and protein-based hormones is that some of these hormones will start out as larger structures, especially these protein-based ones. They can be made of many, many, many amino acids. And so we see terminology like pre-pro hormone or pro hormone. And what we're talking about here is really larger structures that can be broken down into smaller fragments. So a pre-pro hormone, okay, is actually a protein structure that oftentimes contains many copies of hormones or various types of hormones within a larger structure. So for example, if we zoom in on this, if you have the process of translation taking place, translation is where you read messenger RNA and you produce a protein. These proteins can be fairly large. They can be what's called a pre-pro hormone, a large structure that actually can be processed within the endoplasmic reticulum or processed within the Golgi. And what you can do is you can begin to separate portions of this pre-pro hormone structure into functional fragments that are then released into the bloodstream. So a pre-pro hormone can actually be processed within a cell and actually contain possibly multiple copies of a particular hormone, or contain different types of hormones within a single protein structure. So pre-pro hormones can be modified by rough ER and Golgi, and then they can be packaged into vesicles, and then they can be exocytosed into the bloodstream. Some good examples. Here's a pre-pro hormone structure. This particular pre-pro hormone is an example of a protein that's been produced that actually contains many copies of TRH. TRH is thyroid releasing hormone. So this single transcript or single translated protein, you've gone through transcription to make messenger RNA and then translation to produce the protein. This protein, what we call the pre-pro hormone, is actually many copies of thyroid releasing hormone. So instead of making each thyroid releasing hormone individually, your body is making multiple copies of it at once. And then these are other fragments that are cut off from that. And so this is a pre-pro hormone structure. Another term we use sometimes is just prohormone. This is another example. This is the POMC, POMC sometimes as it's referred to. And this pro-opiomelanocortin protein, so this is a long protein that's been translated, and now it's being processed as you go through the ER and the Golgi. And this particular POMC actually produces multiple types of chemical signals. So you've got ACTH, adrenocorticotropic hormone. You've got a adipose tissue hormone, gamma lipotropin. You've got beta endorphin. You may have heard of these. These have like analgesic type effects. So this particular pro-hormone structure actually produces different types of chemical signals. So POMC is another example. And again, they're packaged into secretory vesicles, and then they'll be exocytosed from the cell. And the exocytosis process is a calcium-dependent, energy-dependent process. Another good example of this pro-hormone process is Pro-Insulin. So Pro-Insulin is actually created as a pro-hormone. So you have a larger protein structure. This is the larger protein. This is the pro-hormone structure. The functional insulin is the blue part. So what has to happen is you have to modify and chop off this C-peptide portion. So this will be the functional part. And then this is the non-functional C-peptide being exocytosed out into the bloodstream. And then insulin can go and bind to its receptors and exert its cellular mechanism of action. The C-peptide, though, is released every time you produce insulin. So what's interesting about the C-peptide is that actually is tested for in your blood to see how much insulin you produce. Because insulin is going to circulate and it's going to leave the blood and it's going to go bind to receptors and it's going to exert its effect on body cells, right? So cells are going to respond to insulin. So insulin binds to an insulin receptor and you have cellular mechanism of action. But the C-peptide's in the bloodstream. So you can do blood analysis for C-peptide and actually determine how much insulin your pancreas actually is producing. So that's a nice diagnostic tool that clinicians can use to see how much insulin you produce because of the fact that insulin starts out as a pro-hormone structure. So that is the introduction to the endocrine system. I hope this helps. Take care.